The topic of today's webinar is at the interface of plant and soil, hotspots for soil organic matter formation. Now, before we get started, just a little housekeeping as usual. If you have any questions during the presentation, please type them into the QA box. We will have the time for QA session at the end. And to keep the quality of the sound functional during the meeting, please mute yourself. Now, I request my colleague, Dr. Mohammad Iqbal, to please introduce our guest speaker to the participants.
Thank you, Dr. Shabir. It's my pleasure to introduce our valuable guest today, Professor Carsten W. Muller. He is from the Department of Geoscience and Natural Resource Management, University of Copenhagen. He has his scientific education, diploma in forest science in 2004 from Technical University of Dresden. In 2009, he has his post-graduation in soil science from Technical University of Munich. And 2015, he has done his PhD in soil science from Technical University of Munich. His research interests are soil biogeochemistry, integration of soil chemical, biological, and physical aspects of biogeochemical interactions between soils, plant and microorganisms with focus on the rhizosphere, soil organic matter stabilization and composition as affected by climate and vegetation. Research includes field-to-lab experiments using isotopes and chemical analysis with special expertise in applying spectromicroscopic techniques like SEM, Nano, SIMS, etc. He has currently some key projects. He is a project principal investigator in rhizotraits, rhizosphere traits, enhancing yield resilience to drought in modern cropping systems. It's the title of the title of the project. The second one is the former was funded by the frame of the German Bioeconomical Initiative. A second project is from 2018 to 2021, elucidating the 3D chemical and physical architecture of soil microstructures by combining spectromicroscopic techniques and developing of novel computational approaches. He is also co-PI in joint project with Luxembourg Institute of Science and Technology. He has some valuable, very good, rather very good publications in communications and biology, journals of communication and biology, and other journals which are of very high rating and impact factor. He has a professional experience of more than 15 years. He is affiliated with international agencies like societies and agencies like European Geoscience Union, German Soil Science Society, German Society for Polar Research. Sir, we welcome you to this program. Thank you joining us. This was a brief introduction about Carsten.
Yeah, thank you, Dr. Iqbal, for introducing our guest speaker to the participants. Now, I will request our guest speaker to please start his presentation and share his guest.
Yeah, thank you. Yeah. I hope you hear me well, and thank you for the nice invitation, and also thank you very much for the warm welcome and introduction. I want today speak about the interface of plants and soil. So the background is that most of the carbon that gets into soil comes from the plant. So plants are the major input for soil carbon. And so I want to go with you at the direct interface between the plant and the soil. But to start with, this is the soil carbon map. So this is the map of the carbon over the world, and what you already see is it's really diverse, and we want to go into this in a bit. But what I placed here, and I'm sure that some of the speakers in this very nice series that you brought up, that you organized, we're already raising this. Carbon is the basis of our life. So most of our food comes from soils. 95% of what we eat comes from soils, and the carbon in the soil, this is not just carbon storage, it's also its fertility, it's nutrient cycling, but it's also important, and there we are with the World Soil Day topic, it's biodiversity. And it's also water availability, it's resistance against erosion. So the soil carbon really fosters a lot of soil functions that are crucial for our human well-being. And if we look into what we do to the soil, what we do to the soil carbon, we see that over the last 12,000 years, we were really successful in decreasing this soil organic carbon. So we lost a lot of soil organic carbon due to our soil management, cropland, grazing, all this reduced our soil organic carbon. And if we wanna keep soils fertile, if we wanna produce crops in the future, if we wanna get carbon storage high, fertility and so on, we have to find ways to keep the organic carbon high, or sustain the organic carbon, or even increase the organic carbon in the soil. What drives this organic carbon are several factors. Besides the climate, what you also see nicely in the carbon map with, for example, the Northern Africa, it's really low in carbon, where you have this hot, arid climate. Relief, lithology means parent material. What is the parent material of your soil? Vegetation, land use, I just covered that with the cropland versus grassland thing, and time. And this drives the carbon in the soil. And at the same time, this drives also the soils itself. So the soil type that you look at, and the soils that you grab in your hands, this is all driven by those factors. And today I want to specifically go to vegetation and go to the plant soil interaction in this respect with the carbon stabilization. It's World Soil Day, or it was World Soil Day, but we're still celebrating it, and it's about biodiversity. And what you're looking at here, this is the interface of a plant residue and microbes in the soil. So what you see is fungal hyphae, you see some bacteria attached to those hyphae, and this is the direct interface of a decomposing plant material that gets from plant material into soil organic matter, into soil carbon, and we want to follow up on this in the following talk. What drives carbon sequestration? What drives the sequestration of carbon from the plant to the soil? You could say it's rather easy. So you take photosynthesis, you take a plant, you take CO2, and at the end you will have soil organic matter. And I guess everybody's agreeing, okay, you need the plants, you need the photosynthesis. You might in between also need microorganisms that convert the plant organic matter into soil organic matter, that do the decomposition, and you need soil material. I just, for simplicity, I put minerals here. But it's a bit more complicated than this. If you go to the biology part, you have different plants. Within a plant, for example, if you have a wheat, or if you have a tree of a certain species, you have different rooting patterns. Also for the microorganisms, you have different microbe communities. You have opuscular versus ectomycorrhiza. So there's quite a bunch of, or there's quite a higher degree of complexity in it. But also when it comes to the chemistry, so the composition of the organic matter, be it relatively easy things like the CN ratio, but also the amount of lignin in your plant, or when you think about biochar, or so to say pyrogenic carbon, it's the chemistry that drives how this functions in the soil. And then soil or classical soil science, soil physics, the soil structure. It's the texture, sensile clay, but also the aggregation. How does the soil structure form? And we have to bring that all together if we want to understand the soil and soil functioning as a system. What are the most prominent components in our soil that keep the carbon? This is on the one side, this is particulate organic matter. These are mostly bits and pieces of plant residues, as you see here. But with ongoing decomposition, it's getting more and more microbial. So you have fungal hyphae on top of it. So you see here some fungal hyphae growing on this particulate organic matter. This is this. And the other part is mineral-associated organic matter. And you heard in this, I think yesterday from Ingrid Kögel-Knabner, I guess a lot of results for this part. This is where we think this is relatively stable, persistent carbon pool, carbon that directly sits at the surface of minerals. And in between, for the cycling, you have dissolved organic carbon or dissolved organic matter. And this is the readily available substrate for the microbes. And this is kind of like the loop in between those solids, so to say. If you look a bit closer, for example, here, this is something, soils from Siberia. These are permafrost affected soils from the Lena Delta, directly at the Arctic Ocean. And just to highlight the differences or the shift from the POM, from the plant bits and pieces to this mineral associated organic matter. You see here the CN ratio, carbon to nitrogen, which is relatively high in the plant, mostly plant derived POM. And it goes down to the MAOM, to the mineral associated organic matter. And with it also the decomposition degree. So we have this nice shift between those two fractions. And in a way, the one feeds the other, so via microbial transformation. But it's not separated in the soil. And what you look at now, this is a macro aggregate. You see in bluish color more the mineral sphere. In brownish, this is a particulate organic matter that is occluded within this aggregate, and then you randomly see some organic matter sitting on top. And this is how it looks like in the soil. And this is a macro aggregate larger than 250 micron. And this is what we have in the soil. And the question is, how does this form? How do plants foster this process of aggregation, of the transformation from plant to mineral associated organic matter, or also the formation of particulate organic matter? One first hint for the role of the below ground carbon input, I want to give you here. This is from a work from a former PhD student, Gerrit Angst, where we looked into beach forests in Central Europe, and we see the observed carbon stocks versus the predicted carbon stocks in those soils. And what we see is that root mass and also root plant residues. Root mass is the living root biomass, and plant root residues, this is the particulate organic matter in those soils derived from the below ground input. And we see that these, together with the clay associated organic matter and microbial residues, describe around 90% of the carbon stocks. So that means roots play a tremendous role for the carbon input, but also for the carbon persistence in soils. And if you think about that, if you think about also about the changes that we're going through due to climate change, global change, what is predicted is that tree species richness, for example, will change. And so with that, there might also patterns of the below ground input of carbon change. And that was a reason, or that was the background for one experiment, or for one paper we wrote together with a lot of colleagues from the US. This is a common garden experiment in Poland, where we have 11 tree species, 11 different tree species. And this, based on this setting, we looked into what drives the persistence of carbon in the soil. And we said, okay, the carbon stability can be measured, or as a proxy or indicator of that, we take the soil respiration, we take the amount of particulate organic matter in aggregates, so particulate organic matter that's protected within a soil structure, the mineral associated organic matter, and then delta 15N and 14C isotopes, isotopic composition of the bulk soil. And we tested this against a large set of factors. So, for example, soil chemistry, acidity, pH, or the earthworm biomass, or the leaf and root traits with decomposition rates, and so on. And what we ended up with is, again, that the root traits seem to be much more decisive and important for the carbon stability or persistence, so to say, in the soil compared to leaf traits. So again, here, clear indication, roots play an important role for the carbon cycling, turnover, but also persistence in soils. To just highlight this in the schematic, it's also from the paper 2019 Global Change Biology. If you look here, for example, in this line, this root acid insoluble to nitrogen ratio, and you see there's a direct effect on the delta 15N, something that's often used as a proxy for decomposition, but also the respired CO2. And so you see this direct feedbacks between root traits and soil organic matter stability indices. But this is like the bulk scale, and what we really like to understand is what happens directly at the interface, what happens directly where the plant meets the soil. And for this, we made an experiment that was together with a PhD student, Vera Baumert. The work is in prep right now. She grew European beech seedlings in top and subsoil material. And the background was we wanted to test different parent materials, sandstone, loess, and Pleistocene sand. And what you see here is the increased aggregation. So we clearly see in the subsoils, when we add roots, when we have roots, this has a clear positive effect on macro aggregation in the soil. And if you look into the carbon that sits there, we see, whereas in the topsoil, nothing much happens because topsoils, it seems they are used to a lot of roots. In the subsoils, the macro aggregates clearly increase in the organic carbon storage. And here, just look for the little dots, and for the subsoil and the arrows, it's the fungi. So the fungi get they increase, fungal biomass increases, and our assumption is that especially fungi that are fed by the plant derived organic matter, they drive the soil aggregation. They also drive the carbon allocation into the soil structure and the soil bulk soils that are not directly affected by the plant roots. The question is also, can we follow this directly in situ? And this was a collaboration with colleagues in Switzerland at Agroscope and FIBE. And what the colleagues there, Juliane Hirte and Jochen Meyer did, they labeled with 13 CO2 wheat in the field over a summer, and at the end we took samples from the rhizosphere, and our aim was to follow the 13C from the photosynthesis into the rhizosphere. And those are the samples from there. What you look at here, this is a cross section of a root. So directly the interface of the root and the minerals. This is again the root cell with the attached minerals here. And now this is an image from so-called nanosims, nanoscale secondary ion mass spectrometry, where you see the distribution of the elements. In red, those are your minerals, and in green, this is the root cell, and attached to it, your microbial cells. These are bacteria in this respect. And what you can do from that is, as we introduce the 13C via CO2, you can follow this 13C, and you nicely see the enrichment here in those hot red colors, and then go to this box plot here, and you see, okay, plant cells highly enriched in 13C, bacterial cells in the rhizosphere also, and also we have this this nice outlier here, which means we find the 13C in our soil structures, and here as mineral associated organic matter. So we have the full swing from the photosynthate into these more persistent soil structures, fed by the plant, transformed by the bacteria in the rhizosphere. So to just sum up the roots, roots are crucial for OM input and throughout the whole soil profile, and especially also subsoils play a large role. But of course, classically, it's hard to measure subsoil carbon. But I'm sure there will be more in the future, and several groups working on that. So, and this is also important to be more included in carbon modeling, what the root brings in. Root exudates trigger macroaggregation in subsoils. So extended by the microsphere, extended by the hyphae, who or which are driving this aggregation, and with a assumed increase in rooting and rhizodeposition, that might even play a bigger role in a warm-off or changed, so to say, future. With that, coming to something else when it comes to plants and soil, coming to litter. So we had the roots as a living part, but at some point, both the root will die and becomes litter, but also you have above ground litter, or on croplands, you have harvest residues that go into the soil either by plowing or you leave it there. So, and the question here was, this is work together with Christina Witzkal, a PhD student I'm working with. How does soil texture, how does soil physics determine the formation of organic matter from plant litter? And what we did was, or what we aimed for, having a loamy soil and a sandy soil. And the sandy soil was actually the same loam. We added sand to keep the same microbes in it. And this is how the setup looked like. This is a lab experiment where we had our small soil cylinders in the upper part, mimicking like a cropland. We introduced maize residues. They were 13C labeled, and let that sit for 3 months. And what we saw is when we looked into the microbial respiration, there's a clear higher respiration in the sandy soil, in the coarse textured soil. We have a higher priming, so there's more of the native soil organic matter is eaten up by the microbes because I add fresh litter. And this relates then, if you look into the MAUAM versus POM, in the soil-derived versus litter-derived, this adds to, I have more POM occlusion, I have more aggregation, and that was something surprising for us in the coarser textured soil. It's not a sandy soil, so it's a sandy loam. So, but with a little bit more oxygen availability and a more active microbial community, you increase the aggregation, you increase the occlusion of particulate organic matter, but also the buildup of mineral associated organic matter. Whereas in the more fine or in the final substrate, you have still a larger proportion of free particulate organic matter, so particles that are not within an aggregate. If you look into what the microbes do, and here we're back with the biodiversity theme of the World Soil Day. If you look just at the yellow columns, those are the fungi, and what the addition of the litter does is it boosts up the fungi. And our assumption again here, as for the rhizosphere, those fungi are the drivers for the soil aggregation. And with this, we are again at this aggregate picture, and we did kind of like comparable approach also with this experiment. We looked directly at the litter surface, and with that here you see directly the litter surface. You see the fungal hyphae directly growing on the litter surface, but you also see directly the clay particles on this litter surface. And if you look now with the nanoSIMs and also going for the 13C over 12C, we see it's highly enriched plant, but also the microbial organic matter. And looking into this, we see, highly enriched fungi, and we see this key role for the composition, but also aggregation. And at the same time, we see also this nice shift in the CN. And if you remember back the slide that I showed you from Siberia, shift between POM and MOM with the CN, here it's inversed, but what you see is the increase in nitrogen from POM to MOM. And this is also nicely demonstrated directly at the interface, directly at the microscale between the plant and the mineral-associated organic matter. How far does this reach into the soil? And how is this driving microaggregation? And this is now the final experiment that I want to show you. This is together with Alex Vidal. She's an assistant professor at the Technical University of Munich. And we looked into the decomposition of tree needles, Norway spruce needles, and we used an artificial soil mixture. Nice thing with artificial soil, there's no carbon in it. So no soil carbon, so you don't need isotopic labeling. It's a really nice approach to stay away from isotopes. And we looked into the gradient from the needle, decaying needle, into the soil. Also with imaging, with spectral microscopic imaging, as you see here. So here's the needle, and these are soil minerals, and you go afar. And this is just a representation of images and of gradients. We have day 0, day 14, day 42. This is the timeline of our incubation. We have the needle surface, and then we go into the bulk soils further away from the needle surface. And what you see already with the bare eye is you get an increase in this green, in these green parts here. And the green parts, this is your organic matter. And now getting this as a data plot, as a box plot, day 0, this is the background, and then you go with day 14, day 42, and you see this clear increase in the organic matter distribution. This is the organic matter area in those samples. What you have here also is this microbial residues at the POM surface, directly the interface. This is where the microbes sit, this is where the microbes eat, and this is where they also exude extracellular polymeric substances, which act as a glue for the minerals. And overall, you have this increase in the organic matter, and here again, fungi act as a vector. Okay, to sum that all up, living roots, plant litter, foster the direct formation of soil structure. So we have a direct formation of aggregation via the increased microbial activity. And this soil structure formation is promoting the parallel buildup of more persistent carbon forms. And it's building up both occluded POM, occluded particulate organic matter, and MAOM. And this is not something that's separated. This goes together. Plant residues are nuclei for soil aggregation. There are older works on that, and there's I think this is known for a while, but still it's not right there. People still see it labile as a labile organic matter that just like gets eaten up by microbes, but it's not. It's a substantial part of the soil that plays a crucial role for soil functioning. Mineral-associated organic matter directly forms at interfaces between plants, roots, particles, residues, microbes, and soil minerals, and this is directly happening at the interfaces. With that, thanks to a lot of colleagues. Thanks for all the funding of this work, and thank you for your attention.
Thank you, Muller, for such a nice presentation. Before I would give floor to my colleague, I will just have one query. Could you please tell me how microclimate affect POM stabilization besides microbial activity?
You mean microclimate in terms of at
the rhizosphere level? Yeah.
This is something really interesting question, and I think this is something where we are still a bit puzzled because directly at the rhizosphere, I think there's not much knowledge in terms of how really like oxygen versus water availability is regulated at this spot. And I think that one of the problems is that we with our measurement techniques are not yet there. But and it's also a little bit pointing in two directions. So there's on the one side you have effects that you release more carbon in the soil. You have priming. You have the dissolution of iron oxides that keep the carbon. For example, Marco Kailovite from the US, he's working intensely on this. But at the same time, you also introduce carbon, so you have a gain. And the root also alters the rhizosphere in terms of pH, in terms of water. The mucilage at the root tip alters the water, so the exudation of the mucilage alters the water content. So this all plays together, and I think we are not yet there to fully understand that because we are kind of lacking the measurement tools to get at that point.
And you also talked about root exudates. I just want to know if there has been any research regarding the effect of root exudates on microbial community in case of allelopathic plants.
Oh, good question. I haven't worked on allelopathic plants myself, and I'm not aware. But surely that should be something. That's an interesting point. I'm not aware of work with that, but it certainly is something that would be really interesting to see how the effect is there.
Exactly.
So
exactly.
Yeah. Agreed.
Thank you. Thanks a lot. And now I will request my friend Dr. Iqbal, if there are any questions, he may please point out here.
There are not many queries. Some of the queries have already been answered by the worthy speaker. Still somebody is asking, for example, when we are working on the forest crops and field crops, where the soil stabilization is more.
What kind of crops?
Forest crops. From one side we have forest crops, and on the other side we have field crops. Where we expected SOM more, soil stabilization more?
So it's forest versus cropland, yeah.
Yes,
I mean, it's completely different systems. So it's I think in the agricultural systems, I think the factor of the management might be stronger than the factor of the root itself, but there is work into deep rooting plants, and I think there's classically there was not so much the focus on below the plow layer. So I think in croplands, the potential to gain in the future more carbon is much higher than for forests. If you have long growing forests, it's more you keep the system intact, you keep the management to keep the carbon high. And this is like the natural input and natural turnover. Whereas in croplands, a lot of the croplands are rather low in carbon. And with a little bit a shift, for example, if you have mixed cultures, if you have catch crops, if you have deeper rooting, more intense rooting, this can increase in the cropland your carbon stocks. And this also is especially in the global south, much more important to close the yield gap. So devastated soils or soils low in carbon with changes or with more perspective or more attention to this rooting input, you could gain carbon and with that also gain soil fertility and all the measures that I started with in my talk.
Thank you, sir. One more query is when we compare two things, texture and soil texture and climate, what is the in comparison, which one will be more effective?
So climate texture.
Yes, when we compare texture and climate, which one will be more, for example, in terms of the measuring SOM?
I think, I mean, overall, it's the climate that restricts the plant growth, that dictates the microbial activity. But of course, it's also structure. I would say climate comes first. This is pushing your plant growth, or it's restricting your plant growth. If you have a lack in water, or it's too hot and your plant growth is down, then it doesn't help if you have a nice loamy soil. That means climate is, I think, really ruling. But if you assume you have the comparable climate, then of course it's the soil structure. So as soon as you have a very sandy soil, most of the carbon sits in a free form, and you have a higher microbial activity as soon as you add the carbon, and there's not much that can hold it back. But at the same time, that means if you have those sandy soils, it's even more important to have a management that sustains this carbon. And this is also something classical, or the classical idea or over the last decades was always, everybody was focusing on this persistent and stable carbon. And everybody wants to have that sitting there forever. But actually what we do, or what we want, is a carbon pool that turns over. You want to have the nutrients flowing. You want to have that active, and to sustain the plant growth, to have crops growing, to get your yield. What that means is you have to keep the management, you have to keep the focus on the management that sustains this maybe labile carbon. And this is especially important for lighter soils, sandy soils, where you have the high microbial activity or not much carbon sitting in mineral associated carbon and so
on. One query, sir, here many students are interested in this isotope work, but we do not have a fully fledged laboratory. How much minimum amount we prepare to start a new lab on small scale, sir? It's my personal question to you, because we want to because on this aspect.
I mean, there are several levels to it. So, for example, the nanosims, or this imaging work, this is rather expensive, and one instrument is €3 million. So this is expensive, but you don't want to run that your own. So there are a lot of labs worldwide where you can go and ask for measurement time and for collaboration, and you can do those measurements. On a smaller scale, to do an experiment, for example, with 13C CO2, this 13C CO2 is rather expensive. So such a bottle is 3,000 euro or something like this. But if you plan the experiment wisely, if you have a clear research question, you don't need a huge approach, you don't need a huge batch. Maybe it's a small experimental setting in a greenhouse or in a growth chamber, whatever you have. And the more important is a good research question and a good replication of your samples to get good statistics. And with that, with several thousands euro or dollar, you're already good with the labeling experiment. And then either you establish your lab yourself, you say, okay, I want to be an expert, or I want to go with the IRMS, so the isotope ratio mass spectrometry approaches. Then I think it's in the hundreds of thousands for an instrument. Not sure how much this is, but I guess maybe like €500,000 or something. But there again, there are also possibilities to collaborate, or there are labs that do those analyses that are experienced, that could measure your samples. So I'm a big friend of collaboration, and I'm always a big friend of you know, if there are experts out. I don't run an IRMS. I have colleagues who do this, and I'm collaborating with. So I sent this out, and I'm happy that there are experts doing that, and I would see it like this.
That means if our university asks your university for collaboration in this regard,
yeah. So collaboration is possible, yeah. Or what was this question? Or
is it possible, sir?
Yeah. So it depends on what is like what is on the offer, or what is needed in terms of instrumentation, what we can do, and so on. Yes, of course. This is depends always also on the research question, or on the amount of samples, or what you're aiming for, and so on. Yes, of course.
Thank you very much. This was from this desk. Thank you very much. Thank you.
You're welcome.
Okay. Thank you, Muller, for giving your time. I really appreciate it. Your lecture was really awesome, and we would like to see you in future and have collaborations with you. At the end, I would like to thank my colleagues and my friends and all participants for joining us today. It was really a nice lecture from Dr. Muller. We are getting a huge response in YouTube as well. So in future, we would like to see you here as well when the conditions will stabilize and borders will open. Then we will definitely invite you here, and we would like to have some collaborations. So thank you once again.
It was a pleasure. It was really nice. Thank you for the invitation.
Have a nice day.
You too. Thank you very much.
Before we go, I would request everybody to please open up their camera for a group photograph. Okay, Muller. Thank you. Thanks a lot for joining us.
Thank you.
Have a nice day.